Okay, shall I introduce our next speaker, Megan?
Yeah, that would be perfect.
Many thanks, Sanjay.
Pleasure.
And
Thanks for inviting me.
Welcome, Mimi. So our final speaker just for this first session is Mimi Tamby, who has a Bachelor's in Science in Biomedicine from Birkbeck College and is currently Research Administrator at the Queen Square Brain Bank for Neurological Disorders, which I'm sure many of you are aware of. So Mimi is currently managing a Medical Research Council funded project aiming to build on the facilities at the Brain Bank to offer high-quality central nervous system tissue from patients with neurodegenerative diseases by linking, unlocking, and enhancing the value of unique clinical and population cohorts. So sounds like another quite technical presentation, but welcome, Mimi. And
Thank you. Can you hear me?
Yes, we can.
Yes, great. Thank you. Thanks for having me. Thanks to Megan as well for organising this. Let me just share my slides. There you go. Can you see my slides okay?
Yes, perfect. Thank you.
Great, thank you. So yes, I'm from the Queen Square Brain Bank. My name is Mimi Tamby. I am a research administrator here. And I think this actually follows on quite well from what Sandra was saying about brain tissue, obviously being an essential resource. So I have quite a brief presentation. I'm going to take you through I'll present the Brain Bank and introduce us and what we do. And then I will speak a bit more on the specific project that I manage, which is called the QSBB Neurodegenerative Disease Human Nervous Tissue Resource, which doesn't quite roll off the tongue. Our co-PIs are Tom Warner, which is actually the Brain Bank director, and Zane Yon-Muktane, who I believe spoke at your earlier session earlier this year. So yes, I'll take you through, and then obviously any questions at the end. So the Queen Square Brain Bank for Neurological Disorders holds a unique archive of brains and tissue donated by individuals with neurodegenerative diseases, as well as neurologically normal controls. So it specialises in Parkinsonian movement disorders, including Parkinson's disease and multiple system atrophy, and we hold the national collection of brains donated by individuals with progressive supranuclear palsy. The collection has been developed to include donated brains from prospectively studied people with familial dementias in collaboration with the Dementia Research Centre at the National Hospital for Neurology and Neurosurgery here at UCL. The QSBB also brain banks brains donated by people with dystonia. So the team at the QSBB aim to provide the highest possible quality of brain tissue for neuropathological studies and for scientific research in the UK and worldwide. The large collection of tissue is supported by good clinical documentation, and all material is fully evaluated by QSBB's neuropathologists. Tissue is donated from registered prospective donors, and all, as well as medical records are stored and released in accordance with GDPR. So we receive between 50 and 100 brains annually. Numbers decreased around COVID, and we're slowly kind of getting back up to closer to 100, which is where we kind of sat before COVID. So PSP and CBD donors are recruited through Prospect, which is a study I'm sure you're all familiar with and was also mentioned by Sanjay as well. And if you attended the research information session earlier this year, you would have seen the presentation done by Prospect's coordinator, Riona Fumi. So Prospect is one of the largest observational studies in the UK looking at patients with PSP and CBD. So to become a brain donor with PSP, you'd have to be part of the Prospect study. And I will include their details if anyone's interested in joining that study. So how does brain donation work? So I've kind of split this into the in-life processes and then the post-mortem processes. So in life, we look to inform, consent, and register. So prospective donors are patients generally at the National Hospital for Neurology and Neurosurgery here at Queen Square, and or participants of a research study or a trial. And then we obviously have the non-disease controls, which tend to be family members of our patients. So patients who are seen in clinic by our director, Tom Warner, are approached by the QSBB research nurse, Maggie Burrows, and a conversation will begin regarding brain donation. So these are people that she's seen in clinic a few times, a rapport has been built, and Maggie offers information and advice on tissue donation and support to patients and their relatives, as well as to clinicians. So patients will be sent away with information sheets and encouraged to have a discussion with their families and the next of kin. If they decide that this is something that they do want to be a part of and they wish to join the registry, they will be consented in clinic by Maggie at a later date. So no one has ever consented on the day that the information is first given to them. They're given the information, asked to go away, have a think, have a discussion, and then if it's something they want to pursue, we would then consent them in clinic. So donors from research study groups will have these same conversations, but with the study group's research nurse who would have been trained by QSBB staff. Once a donor has signed the consent form and decided that they're going to participate, they are then registered to the donor database by the QSBB administrative team here, who will then send them out a welcome pack. So the welcome pack includes a donor card, a self-assessment form where you are asked things about your symptoms, the duration of your symptoms, and any kind of medication that you're on, amongst other things. You'll also receive a copy of the consent form that you signed and instructions on what to do at the time of death. So once a donor passes away, the family and next of kin will notify QSBB and arrange for verification of the death and for the funeral director to take the deceased into their care after verification. So that is normal practice regardless of whether or not a brain donation is taking place. The QSBB admin team will liaise with the family, the GP, the medical examiner, funeral director, and a local mortuary to set up a brain donation. Any additional costs incurred in relation to the donation is, of course, met by QSBB. Tissue is then transferred to the QSBB offices by courier. The admin team here provide an on-call service during evenings and weekends and liaise with relatives, hospital staff, funeral directors, and couriers to ensure the careful donation of the brain with minimum distress to families. So slightly further down the line, the next of kin will receive a call from Maggie, our research nurse, to discuss the histopathology findings. They'll then receive a hard copy of the pathology report if they request one or if they want one. At that point, if they then want to discuss that further with the treating consultant, that's also something that we can arrange. So tissue is stored as fixed blocks, and the majority of cases also have flash frozen samples available. Flash frozen material has excellent histological preservation and is suitable for in situ hybridization and immunohistochemistry. So tissue can be provided as the slide mounted sections or as small frozen blocks for neurochemistry or DNA and RNA analysis. Case controls are matched for age, sex, and postmortem delay and are supply blind. Clinical data relating to the tissue supplied is also made available on an anonymized basis. So QSBB also has a digital pathology resource, which is in part funded by Aligned Science across Parkinson's ASAP through the Michael J. Fox Foundation for Parkinson's Research, and it's available for viewing representative diagnostic slides on our website. The QSBB Tissue Request Committee review all requests with approval based on the researcher's expertise, track record, and the merit of the research plan. So if necessary, additional information may be sought from the researchers and a new request submitted. After approval of a tissue request, the researcher and the researcher's institutions agree to and sign an MTA, which is a material transfer agreement, prior to the supply of tissue. So just to give you an idea of sort of the amount of samples that we're sending out, in 2019, 29,000 samples were sent out to research groups, both here in the UK and internationally. And so far this year, we have signed 18 MTAs. So that was just a brief introduction to the QSBB. Now I'll take you through the project that I work on, which is the QSBB Neurodegenerative Disease Human Nervous Tissue Resource. We've been funded by the Medical Research Council for 3 years, and we've just completed 1 year. Our main objectives are to build on the existing resource here at QSBB and to increase the quality of the tissue by recruiting 650 new donors from 8 clinical cohort groups with linked in-life data, by decreasing the time between the donor passing away and the donation arriving at the QSBB, which is known as the post-mortem interval. Currently, that's at approximately 3 days, and we are hoping to get it as close to 24 hours as possible. And by improving the value of the tissue, by genotyping the entire collection, as well as creating annotated digital slides and digitizing clinical records for the entire collection as well. We're also creating an easily accessible portal where researchers from academia and industry can search for available tissue and also see what associated in-life data is available. And finally, we want to increase the diversity of our donors by engaging with patients and understanding what the barriers are to brain donation and how we can go about removing those. So these are the 8 clinical cohort groups that we plan to onboard. So the first is the Rare Dementias group, which is at the Dementia Research Centre here at UCL. It includes over 200 cases of rare dementia presentation, particularly young onset and familial Alzheimer's disease, with extensive clinical, genetic and imaging data. Second cohort is a cohort of sporadic and genetic frontotemporal dementia cases, also at the Dementia Research Centre here at UCL, and also with extensive clinical, genetic and imaging data. The Vision in Parkinson's study is a Wellcome-funded study with Parkinson's disease followed up with longitudinal clinical neuroimaging, plasma and genetic data. The Rhapsody study is a group of patients with Parkinson's disease and their relatives with a focus on those that carry variations in the GBA gene and those with Gaucher's disease, which is a common risk factor for Parkinson's. This group has long-term clinical and biomarker follow-up data. The motor neuron disease cohort group are undertaking serial clinical follow-up with additional imaging, blood, cerebral spinal fluid samples taken to identify traceable biomarkers. And then we have the Down syndrome cohort, which is a small group of volunteers for which there's extensive clinical, cognitive, and wet biomarker data available. People with Down syndrome have a significantly higher risk of developing Alzheimer's disease. And then we have the 2 population cohorts. So the 1946 birth cohort is the longest running British birth cohort study with serial data on cognition, sociological factors, imaging, fluid biomarkers, and genetic data. A subset of that group is the Insight 46, which is a neuroscience study that we are recruiting from. And then finally, we've got Predict PD, which is a very large study following 10,000 people who provide clinical data, a smell test, and perform simple online tests to identify those at higher risk of Parkinson's disease. So we aim to recruit those at the highest and the lowest risk groups within that cohort. So as I went through all those groups and I mentioned kind of what the in-life data is available for each of them, this is something that we plan to make available through our online portal, linked with the tissue samples once they are received. We are also planning to link to the tissue held at QSBB. We'll also be planning to create new fibroblast resource from these cohorts to provide a unique live cell tissue resource of 150 new disease control cell lines. This resource will be linked to the IT portal and will permit searches and requests. So the research value of the collection will be increased by addition of genetic data. All cases have now been genotyped and will soon be made accessible to researchers. Digital slides have now been created for over half of all cases, and digitizing the entire archive will enable quantification of misfolded protein pathologies across different disease cohorts and multiple brain regions in an automated, consistent manner using machine learning-based digital pathology quantification algorithms. These are currently being developed. Automated quantification of misfolded protein pathologies provides an unbiased detail about a certain pathology load in a region of interest and therefore enables large-scale, consistent pathology quantification for integration with other multiomics, such as genetic data. Similarly, digitizing all patient records will allow us to create a comprehensive clinical database that accompanies the pathological resource and permits AI or machine learning-based algorithm to test hypotheses across the pathological and clinical resource. So our second objective is to create this online portal. We are collaborating with Dementia Platforms UK to create an easily accessible portal for researchers from both academia and industry to search for tissue and then be able to see what in-life data is associated with the samples and what is available. The pilot portal will link to work being done by colleagues in Newcastle and at Oxford. With the ultimate aim being to provide a portal with the ability to expand, to incorporate data from other biobanks in the UK, and also to link to other associated data, such as NHS records. So, as I mentioned earlier, we aim to reduce the post-mortem interval from 3 to 4 days to 1 to 2 days by targeting the main hurdles to timely donation. So that is arranging and performing the post-mortem and transporting of tissue back to QSBB. So currently we rely on local mortuaries and morticians to carry out the donations, which can sometimes cause delays depending on how busy, how willing or how able they are. So to solve this issue, we are currently recruiting our own anatomical pathologist technician who will be mobile and able to travel to the donor's location, access mortuary space in a local NHS or private mortuary to undertake rapid tissue retrieval and arrange for the transport of tissue to QSBB with minimal delay. So once we've received enough donations through this sort of fast post-mortem pilot, we will then perform a pilot study on the effect of the PM interval on RNA and tissue quality. The anatomical pathologist technician will also be able to obtain additional tissue samples to address wider research needs. So obviously this will only take place if the donor has consented to it. And depending on the diagnosis, they will include skin, skeletal muscle, peripheral nerve, heart, and gut tissues. And finally, we would like to diversify the donors that we have on our registry. So we're aiming to understand how we can best overcome barriers to donation and to achieve a registry that is more diverse, which in turn will facilitate research outputs more representative of the broader population. So currently, fewer of 10% of our donors come from a non-white European background. We are working to engage with patients and the public to understand why that is. Obviously, in some cases, there are cultural and religious issues that prevent people from considering brain donation. But we believe that through kind of engaging with the public and with patients, we'll be able to overcome these barriers and ultimately diversify the donor registry. So we held our first PPI event at the end of last year in November with a group of patients with Parkinson's disease. And we did get some really interesting feedback from them. A lot of them were very engaged and aware of research and participating in trials. So sometimes you are picking from the same pool of people. So what we're doing is trying to really kind of cast our net really wide to get opinions from those who maybe aren't so aware of brain donation or research in general, to kind of understand where they're coming from and kind of what we could do to better inform them on what it is we do here and the benefits of brain donation. So on that note, this is my contact details, my email address is here, so please do feel free to get in touch with me. We will be holding another session before the end of this year, and I'm more than welcome to receive your name and details if you're interested in volunteering for that, or even just if you have any kind of feedback that you'd like to kind of provide, I'd be more than welcome to receive that. This is also the contact details for prospects. If you're interested in participating in that study, please do feel free to get in touch with them directly.
Thank you, Mimi. Do you want to just stop sharing your slide?
Yes. There you go.
Excellent. So it sounds like quite an ambitious investment from the Medical Research Council into improving and digitizing the quality of the data we hold. Are there other locations in the world who have comparable brain banks, or are we looking to kind of be a leader in this?
So we are looking to be a leader in terms of the portal that we're putting together. We are kind of looking to be a leader in that, in the way that we're linking the tissue to the in-life data, as I mentioned. So as far as we're aware, there isn't anyone else quite doing it in that way. So we're kind of hoping to be pioneers in this. And as we continue to grow, we continue to onboard different brain banks. So then the portal will give you access to their data as well.
Excellent. Okay, Megan, I can't see any questions from the audience in the chat.
There's one in the chat asking if you are looking for those with normal brains or no known neurological conditions to donate, which I would assume is a big yes.
Yes, absolutely. Yes, we're always looking for healthy controls as well. If you are a control and you are interested in donation, you can make your way to our website. I will put the details. I should put that on the slide. I'll put the details for our website in the chat so you can access it that way.
If you send
yes,
them to me, I can
always
pop those
looking.
in there.
Okay, perfect. Thank you. Yeah, always looking for healthy controls as well.
It's just worth saying, Megan, I'll put a link in the Q&A or the chat as well, that there are brain banks in other part of the UK as well. So
Yes.
if you're on this call from outside sort of London and the home counties, there are other options if should you wish to donate your brain in both Scotland and England. I don't believe there's one in Wales or the North of Ireland.
There isn't, no.
Okay, so yes, your traveling physician who will be going, it's just in the south of the UK, is it?
Yes, unfortunately we are. So this is one of the issues that we have, which is why we're kind of looking at this fast PM. So unfortunately, the cost of couriering a brain to us at the brain bank is very, very high. It's normally now roughly about £5,000 per brain to the cost of kind of retrieving a brain. So that's the post-mortem and getting the brain to us. So we are sort of limited to an area that is kind of not too far from London, just because the distances involved mean that the costs obviously increase. But we are hoping now that we are recruiting this anatomical pathologist technician, it kind of will remove the courier aspect of it, because they will be the one doing the transporting of the brain. So once we have that person in place, we'll have a better idea of kind of the distances we're able to cover.
Okay, no, interesting to know, because the PSP Association already is providing some funding to support the transport of brains. I think it was in Scotland, but it's the sort of area that you can imagine donors maybe being interested in, supporting such a valuable investment, isn't it? So
I think compared to the costs that Mimi's talking about, we cover purely transportation, not the other elements of retrieval in Scotland, because they already have funding for that. But yeah, we do, because sort of Highlands and Islands, can be quite difficult. The cost for actually just bringing the brain. Yeah.
No,
and of course, we're always happy to receive anyone's support. There is a donation page on our website as well, so I'll pass those details on to Megan as well.
Yeah, no, thinking in terms of donations that can have a real impact, looking forward. So very, very glad to hear there's so much investment going on in this, because my background's in technology, so I can see the value of getting this data digitized going forward. So Excellent. Well, I don't know why I can't see questions in the chat, Megan, but if there aren't any others, I'll
check. There's no more questions in the Q&A box, but I think a question I might have is, from one person's brain donation, this might not be a question you could answer, how many projects could that one brain go to help? Oh, you're on mute at the moment.
Sorry, I've muted myself.
There is.
I don't know exactly. I can find out for you, but it could be within the tens, I'd say, probably.
That's, yeah, incredible to think of your, the donation being able to go and help not only projects in the UK, but projects all over the world as well.
And once the data is digitized, that's limitless, isn't it?
Yeah, exactly. Which is obviously the benefit, because tissue is obviously a finite resource, but once we've digitized and the slides are available, it is there forever.
Very, very interesting. Well, many thanks, Mimi. I suspect we'll see you back if you've got a couple more years to run on this grant. But
yes, thank you. Thanks for having me.
Exciting things happening. Okay, so Megan, do you just want to set people up for the second half?
Yeah, so we'll be taking a quick break, so go grab a coffee, a cup of tea. We'll be back at 12 o'clock to continue with the last two speakers.